Hi everyone, today I'm going to go through the alleles crossing over to the other side lab. It looks like this on the front. It's what we had started in class, and we close read through this whole background information part, and we answered pre-lab questions. And then on the day that we actually did the lab, I know that many of you were out, and some of you might still need to refer back to this video so that you can be able to answer the questions that are on the back side or the post-lab questions. There are 5 of them. Also, this is a great video to reference anytime that you want to look back on meiosis and see how crossing over contributes to genetic variation and how it is that we have genetic diversity. I'm going to go through each step of meiosis, or this lab, alleles crossing over to the other side, and while I do it, I'll show you here on the paper with these models what's happening. So we're going to assume that interface has already happened, that the genetic material has replicated. You may or may not remember actually having to label all these beads in order. So there are matching beads A, B, C, D, E, F, G, H, and they correspond to the types of alleles for each of the genes. So we had capital letters on one chromosome and then lowercase letters on the other one. Some of you may have made it more challenging for yourselves and actually mixed up the letters a little bit, and that's okay. I just have one chromosome with capital letters and one chromosome with lowercase letters. Alright, so we're going to start with meiosis 1. Meiosis 1 starts with prophase 1. Prophase 1, we have both of these wire models inside of this cell. So the paper represents the cell, and these are our chromosome models. We're also going to simulate 3 events of crossing over by exchanging the pink and white beads with the same letter on them. I'm going to start with just a simple one. I'm going to exchange the pink A with the white A. So now that is one event of crossing over. I'm going to do this 2 more times. I'm going to take the C off and exchange the C. Oops. Oh, I put that B on backwards. Oh well. Okay, and then let's take the H beads and exchange those H's. Okay, so that was 3 events of crossing over. Now my chromosomes here are not identical anymore. They have genetic information or variation that has changed, and now they're more diverse. We're going to move on to metaphase 1. We're going to line up the chenille wires in the center of the cell. The green is going to be on the left and the white on the right. Anaphase 1, we separate the chromosomes by moving the green to the left and the white to the right. Telophase 1, we tear the paper in half. This simulates that the cell has now divided into 2, and each one now contains about half of the genetic information that we started with. So we started with 2 chromosomes in one cell, and now we have 1 chromosome in each cell. So that's about half. We are going to start with meiosis 2. Meiosis 2, in the packet it talks about an interphase 2. Interphase 2 does not always happen. The DNA doesn't always unravel. So I'm just going to kind of skip that and move on to prophase 2. In prophase 2, if the genetic information or these chromosomes had unraveled back down into like strands of DNA or what we call chromatin, they would condense again back into chromosomes. So technically prophase 2 has already happened. And then we have metaphase 2. So we're going to line up the chromosome models in the center of each cell, but this time they're about 90 degrees from their original position. So this turns this way 90 degrees, this turns this way 90 degrees. And then anaphase 2, we separate the sister chromatids. So we're unraveling these wires. And then these 2 wires, spindle fibers would attach to them and then pull them to opposite poles of the cell. So I'm going to simulate that by just moving the sister chromatids to opposite poles of the cell. And then in telophase 2, again, the cells split. So I tear the paper in half again. So that's the second division and the end of meiosis 2. Now, you can tell just by looking at these four things that each one of these cells is all different from each other. The genetic information that is in here no longer matches the genetic information that's in here. It does not match the genetic information in here or in this one here. So these cells are all unique. These cells are haploid, they're gametes or sex cells, and they would go on to contribute genetic information during reproduction, and the offspring of these would inherit the traits that are on here. So this is why you do not look like your parents, exactly like your parents, because you don't have identical information on your chromosomes anymore, because crossing over took place. And also, you don't look like siblings, cousins. You're all unique in your own way because these haploid gametes are all unique in their own way due to meiosis and crossing over. I hope that this helps you guys out. If you have any questions, let me know.